The overall goal of this procedure is to study DNA methylation and gene expression changes upon early life stress. This is accomplished by first subjecting newborn mouse pups to maternal separation to induce early life stress. As a second step, brain areas of interest, here the PVN, are obtained by in loco microdissection, and DNA and RNA are isolated simultaneously from a single tissue punch. Next, the obtained DNA is bisulfite treated, and the region of interest is amplified by bisulfite PCR. This is then ligated into a vector and transformed into bacteria. Successful transformations are identified by marker expression and PCR fragment size. And lastly, bisulfite sequencing is used to assess methylation status.
The main advantage of this workflow is that it allows convenient analysis of epigenetic programming in response to environmental stimuli.
This method can provide insight into epigenetic programming by early life adversity. It can be also applied to other systems wherever methylation and gene expression are analyzed in highly tissue-specific environment and wherever tissue availability is limited.